Alright, let's have a little more word on the blood vessels. So we have the renal artery feed in through multiple divisions blood into the glomerulus, which is the capillary network. The afferent arteriole feeds it in to the glomerulus, and from there efferent arterioles lead the blood away from the glomerulus, and from that first capillary network. But then what happens is it goes around and hangs around in a secondary capillary network, running around into the medulla, forming a web around the renal tubules, the loop of Henle there. So you can see that here. You can see the renal tubules come down, which are the loop of Henle down here. And then you have that capillary stuff hanging around there. And that's known as the vasa recta. So that's a secondary capillary network. And from there then, because these are areas where a lot of the substances go back from the tubular system into the bloodstream, from the vasa recta then we will leave the blood into small veins, and then we get into arcuate veins, into local veins, and then back to the renal veins, into the vena cava, bring the blood back to the heart. So what we need to remember from there is, besides those little artery names, which are not that big of a deal, what's more important functionally is that we have a first capillary network where all the blood, or a lot of the blood stuff, goes into a filtrate, into the tubules, and then we have a secondary capillary network that hangs around the tubules and recollects or reabsorbs most of the substances that were filtered out the first time. And then of course, remembering back, we also secrete substances from the blood into the tubules at that point, things that don't just automatically go through. Alright, I know. So when we look at the glomerulus a little bit closer, we have the renal corpuscle, which is the whole structure itself, the Bowman's capsule, as well as the glomerulus, the capillary network. And so let's look at the capillary network there. When we look at that, this is one of those capillaries. We see these weird cells that sort of cling to those capillaries, and those are known as podocytes, and those become the inner layer of the Bowman's capsule. The Bowman's capsule is a double-layered membrane. It has an inner layer that hangs around and clings around the capillaries, and an outer layer, and that makes that shell, the structural shell of the Bowman's capsule. And that here becomes the filter. So the podocytes are growing close enough together that their little slits are not too big to let cells and things of higher size, large molecules, through. So they leave those behind in the blood, because that becomes an important when we want to bring substances back into the blood. Because when we have particles left behind and their concentration gets denser, they become water attractor. So they always pull. That's an osmotic pull that we have. Particles can always create that water pull effect. And you always think about that when you cut a tomato and you put salt on top. After a few minutes, you see water droplets appear. The salt attracts the water and pulls water with it. That's an osmotic pull. All right. So what we get here is a glomerular filtration barrier when we have these podocytes go around the capillaries, and that's about a square meter we have in each kidney. I think it's each kidney. And I look at the glomerular filtration rate. That's measuring the kidney's ability to excrete, and it's about 120 milliliters a minute. Measures how to filter. What we can do, we look at the kidneys as a mechanical filter. It pushes the blood coming through and pushing it through those podocytes, and here comes in, pushes it through the podocytes and gets collected in here. So that is done via hydrostatic pressure. That's a passive event. So a lot of stuff gets through that way. We also have, and so that's a passive event, depends on the blood pressure in the capillary tufts and the glomerular surface, filtration surface, of course. And we also, though, have an osmotic pull that happens because the large molecules are left behind. So they will pull back, and that's the blood colloid osmotic pressure. Look at the measure that even. The hydrostatic is about 55 millimeters of mercury. The osmotic opposing is about 30 millimeters of mercury. And then what we have as, which is kind of weird the way we have to think about it though, as the pressure pushes out, we have the capsule that's firm also exerting a pressure back in, a hydrostatic pressure. And that's about 15 millimeters of mercury. And so we end up with a net pressure, a net outward flow, what they call here 10 millimeters of mercury. You don't have to memorize these numbers, it's just that way you explain them right there. And so that's how that works, and so we can measure that. Then, now how we get from filtrate to urine is the next question. So the reabsorption process is a good example of how active and passive processes are closely interrelated in the body. That's right, we have a play here. We have sodium potassium pumps that are located in the basement membrane of the tubule epithelial cells and actively transport sodium back into the bloodstream. So look at that, we have sodium potassium. And as I said before, with sodium follows water. So table salt sodium chloride. So with the sodium follows the water. To maintain the body's sodium concentration, we excrete all salt eaten via the urine. So that's where eating the salt becomes a problem with the high blood pressure, because water and salt work together. So more salt we have, and of course if you chronically eat high salt content, the body will raise that salt content and it will bring water in, more water in. So we retain more water, and that raises blood pressure. Anyhow, 90% of the salt, sodium chloride, and 60-70% of the water are reabsorbed in the proximal convoluted tubules. That's right here, see, that's where we see this sodium going out, proximal convoluted tubules, and in the loop of Henle. That's down here where the loop of Henle is. Alright? We concentrate on the sodium, not the other stuff. And then fine-tuning of sodium balance is mainly taking place in the distal convoluted tubules. Because what we do there, we have aldosterone, which is promoting sodium reabsorption, and also potassium excretion and so forth. But we worry about the sodium right here. That's the main one for us right now. And it does that with the aid of renin. We'll talk about that in a minute. We have a slide on that. So renin and aldosterone work together. And so the aldosterone is reabsorbed. No, the aldosterone makes the sodium being reabsorbed, and that will bring more water with it. But then how much urine we actually pull out or excrete will be more finely tuned through ADH, and that's the antidiuretic hormone. That comes from the anterior pituitary gland. If you remember, we had the pituitary gland in the endocrine system. And what that does, the antidiuretic hormone, which I call the anti-P hormone, which makes it easier to understand the processes, for me, opens aquaporins in the collecting duct. So that's when right before we get into the pelvis and then the ureter, so as we finish up the process, we have aquaporins that, aqua is water, that lets water flow through the interstitial fluid into the bloodstream. And because the interstitial fluid is hypertonic, it's very salty, so to speak. It will flow straight through into, it will be pulled out when these aquaporins are open and go into the bloodstream. And as osmotically active particles like sodium in the blood plasma rise, osmoreceptors in the hypothalamus will work and stimulate the ADH release from the posterior pituitary gland. Did I say anterior before? It's posterior. So the hypothalamus and the pituitary gland work very closely related in controlling the body function. So that's interesting there, where you have really the hypothalamus doing a lot of work in terms of controlling things. Look at that. Here's the renin-angiotensin-aldosterone mechanism. So we have an apparatus called the juxtaglomerular apparatus. And juxta means next to. And so that's a place right next to the glomerulus where we can measure systemic and local blood pressure fluctuations. And when the blood pressure falls in those areas, the renin is secreted by that apparatus, and then that starts a cascading effect leading to finally aldosterone activation. But it also goes through, have some factors from the lungs, and it goes through an angiotensin 1 and 2, and the 2 is a very potent vasoconstrictor and a thirst creator, so we drink more water, and then the aldosterone is activated, and then that goes and activates the sodium reabsorption from the filtrate, which then brings water with it and raises the volume in the blood volume further. And so that raises blood pressure, of course. And so that, if we have a leak, we want to make sure we don't leak out, so we have to make more. You always, in terms of how the function is, you kind of, in those things, if you think of emergency situations versus long term, then you know why the body does that. In an emergency situation, we want to maintain blood pressure when it falls down. Of course, in our society, our life, our modern life, we have more long-term problems where we keep blood pressure high over time because we don't have as many of these emergency situations anymore. But there you have it, Renin-angiotensin-aldosterone mechanism. And that brings us to the urinary tract. The wall of the urinary tract is composed of smooth muscles and lined with transitional epithelial tissue. There's a typo up here. Transitional epithelial tissue right in here, then that's the bladder, that's transitional. So what that means, transitional means you can stretch the tissue. You have all kind of ability. You can have the cells be cuboidal, columnar, or stretchy, squamous type looking things. From the renal pelvis, peristalsis moves the urine along the ureter, entering the floor of the bladder. So if you look at the bladder here, that's the bladder. These two holes, that's where we get the ureter coming in. Ureteral orifice, an orifice is a hole. And then the urinary bladder lies in one's true pelvis, so that's right behind the pubic bone, immediately there. It narrows downward a little bit here, narrowing, coming down to a funnel type. And it transitions sort of into a urethra. And then the urethra is where the urine is voided out into the real world. We have an external sphincter here that controls if we want to go pee or not. And we have an internal sphincter right here by the opening. And that area between the two openings on the top and the opening on the bottom is known as the trigone. All right. Trigone. Look at that. I think that's it. Well, there you go. I hope that helped.